So we're finally down to our last lecture, and this is a rather long one. 105 pages, in fact. That was just purely by accident. So this last lecture of Bio 105 is going to be mostly review. Most of these pathogens are things I've talked about before, and most of the concepts are things we've talked about before. But what I'm going to be doing is going through these pathogens again and a few new ones, and this time breaking them down by body system. So we'll look at infections of different parts of the body. And there's a lot of parts we're talking about here. So I've broken it down into these 6 parts here. First, we'll talk about body surfaces. So we're talking about the skin, but also conjunctiva. And then part B is going to be the urogenital system, then the respiratory system, digestive system, nervous system, and finally the circulatory system. And in part F, we'll also look at systemic infections. Systemic means system-wide, so body-wide. So as you can imagine, if you have a pathogen that gets into the blood, it can travel anywhere in your body, and you can have a body-wide infection, which can be very, very serious. So it's possible that you might start off with a fairly minor infection of the skin, for instance, but that pathogen moves deeper into the skin, finds its way to capillaries and gets into your circulation, and then that can become far more problematic. Okay, so starting with body surfaces, and we'll spend quite a bit of time on this. Makes sense that there would be a lot of different skin infections because, of course, your skin is your main defense against the environment. It's in contact with your environment. And it's a huge surface area. It is the largest organ of your body. So the skin is our main protective layer, as I've mentioned, and it's also the body's largest organ. It's a huge surface area. It tends to consist of dry, keratinized, dead cells. So in some areas, you can have 40 or more layers of these dead cells that make up the stratum corneum, the outer layer of the skin. And because they're dead, viruses can't reproduce inside them. Bacteria have a hard time feeding on keratin, which is a tough protein. Also, they may contain some oils that are fungostatic. That means they limit the growth of fungi, but they may contain some oily substances that also limit the growth of bacteria. And then on the surface, you have the microbiota or microflora, which are the communities of bacteria that are competing with each other and competing with any pathogens that might arrive at the scene. So they're stopping those bacteria from gaining a foothold and taking over. We also have the conjunctiva, and the conjunctiva is the layer of tissue, and this is a slippery layer, it's not a dry layer, that covers the outer portion of the eye and also lines the inside of the eyelid. So it's a continuous layer. It prevents things from getting behind your eye, back into the orbit of your eye. Conjunctivitis, that is an infection or inflammation of the conjunctiva. So if you have bacteria that are damaging this layer, that's going to result in increased blood flow to the site of the infection. You have very fine blood vessels in the conjunctiva. They're normally fairly small and not that obvious, but if you are fighting an infection and they're inflamed, then they become much larger and more visible, and that gives the tissue this pink color, hence the name pink eye. So that's conjunctivitis. If you see itis at the end of a word, it means an inflammation of whatever precedes that itis ending. So conjunctivitis is an inflammation of the conjunctiva. And there are a number of bacteria that live on the skin that could potentially cause problems. Again, you've got billions of organisms living on your body. They're generally not causing you much trouble, but any of them could potentially cause an opportunistic infection. So Staphylococcus aureus is something that appears several times on this table here, and that's because it is problematic in some cases, but we have some other bacteria species as well at the top, and we'll come back to some of these. We have several viral species, quote unquote, that can cause infections of the skin and damage of the skin as well. We've talked about varicella before. That's one of the herpes viruses, and we've talked a little bit about smallpox. We'll come back to that. And we'll talk about rubella and rubeola very briefly as well. We have fungal infections that can cause problems, usually not severe problems for healthy people. So we do have fungi that can cause thrush and yeast infections. We have fungi that can cause athlete's foot. But in people that are immunocompromised or have other pre-existing conditions or other pre-existing infections that their immune system is dealing with, fungi can be quite problematic. So fungi can be life-threatening in some cases, particularly if you have diseases like AIDS. If you have an HIV infection, that can dramatically affect your immune system, and you might not be able to fight off a fungus that you normally can keep at bay. So on the surface of the skin, we have a community, a little ecosystem of bacteria, and there are quite a few that can potentially cause problems. So we have the Propionibacterium acnes, which, as you might guess, is one of the bacteria that causes acne. It's one of the most common causes of acne, and it is a gram-positive rod. And then we have a couple of Staphylococcus species that we've talked about already. Remember, Staphylococcus means that they grow as little spheres and they're clumped together. So we have Staphylococcus epidermis. It can cause some minor skin infections, can be problematic for some people that are immunocompromised. And then we have Staphylococcus aureus. Again, not something that normally causes problems in healthy people, but it can. It's more likely to cause serious problems than these other bacterial species. So the reason that S. aureus is of particular concern is because it has a number of virulence factors. And these are characteristics that make the bacteria more likely to cause physical harm to your cells and to your body. First of all, S. aureus, like some of the other skin bacteria, is salt tolerant. So salt is released from your skin. It has a few functions. I mean, one of the reasons you do that is to get rid of excess salt, but also the salt is going to limit the growth of most species of bacteria. So most species of bacteria don't grow well in a salt environment. It might not kill them, but it limits their rate of growth. But S. aureus is tolerant of salt conditions, and we did a test of that in the lab. If you remember, we grew bacteria species on mannitol salt plates, and S. aureus and S. epidermis did quite well under those conditions, whereas things like E. coli did not. S. aureus can form slime layers. That's where we have multiple layers of bacteria that stick to each other. They secrete this protein that kind of acts like a glue to stick them together. And the cells that are in the middle of that slime layer are protected from antimicrobial substances and oils that the skin produces. Some strains of S. aureus can carry an enzyme that destroys penicillin and other antibiotics and other substances that are produced directly from the skin. And there are a number of toxins that are produced by S. aureus. Many of them are just waste products, so they're just byproducts of their metabolism. Some of them are produced specifically to target cells and destroy cells. So we have proteins that can clot blood. We have enzymes that can break down connective proteins. And this is another thing that we looked at in the lab in Quesnel. So if you take media and you add gelatin to it to make it a solid, gelatin consists of collagen. And if you add S. aureus to a tube of that, the S. aureus will break down the collagen into little pieces and the contents of the tube will liquefy. So that's a gelatinase test. That shows that we're dealing with a microbe that can potentially break down collagen. That's important because a microbe that can do that can break down the extracellular matrix that surrounds our cells and sticks our cells together. So S. aureus is problematic because in some cases it can dissolve the collagen between cells and maybe dissolve the collagen at the base of epithelial layers, the collagen that makes up the basement membrane. And if it does that, it can get deeper into the connective tissue and maybe into the blood flow as well. So one of the more minor conditions that can be caused by S. aureus and other bacteria that live on or in the skin is folliculitis. and folliculitis is an inflammation of the hair follicle. So again, itis means inflammation. So what happens here is we've got an infection of the follicle, which is at the base of a hair. Hair is dead, it's just made up of protein and it's being excreted by this follicle. But if we have an infection, we're going to have increased blood flow to that area, and that's going to produce reddening and a bit of pain as well as the tissue swells. We may have a pimple forming, and the pimple is going to fill up with pus, and the pus is dead cells and debris. So white blood cells that are coming to the area in the increased blood flow are going to gobble up bacteria. They can only gobble up so many before they die. And the reason they die is they run out of lysosomes that are packed with enzymes to break down those bacteria. So the white blood cells will sacrifice themselves and die after they've killed a few of the bacteria. The bacteria, of course, are being destroyed. They might be destroyed in the interstitial fluid by components of the complement system. So we have this pus that forms that's dead white blood cells, dead bacteria, and bits and pieces of debris from that war that's going on. We can also have something called a sty forming, and this is where we have oil glands that become infected. So oil glands, sebaceous glands, are usually associated with hairs. And these are particularly noticeable around the eye. So the eyelashes are common places for you to get this fairly large swelling called a sty, but again, it's caused by an infection by bacteria. Quite often that would be S. aureus responsible. Another condition caused by S. aureus is impetigo. And with impetigo, you have these reddened patches of skin. Again, that's because of increased blood flow to the area. And we get these large areas that can fill up with pus, and then we get this crusting over that occurs that produces a rather noticeable scab on the surface. Far more common in children because, well, let's be honest, children are not perhaps as hygienic as they could be. And they're more likely to scratch at a small wound or a small infection and make it larger by introducing Staphylococcus aureus into that cut or into an area that's already damaged. We can have larger boils that form as well. So here we have fairly large pockets of pus, little blister-like pockets of pus that are occurring quite deep within the skin. So these are conditions that can not only damage the epidermis, but they can damage the dermis as well, the deeper layers of the skin. It's also worth noting that impetigo is very commonly seen in children directly under the nose. And again, the reason for this is because a lot of people have permanent populations of S. aureus that live just within the nostrils. Another more dangerous condition caused by S. aureus is staphylococcal scalded skin syndrome, SSSS. What's happened here is that Staphylococcus aureus has gotten deeper into the skin. So it's gotten into lower layers of the epidermis, maybe into the dermis as well. And some S. aureus will produce a toxin that causes layers of skin to detach from each other and peel away from each other. So we have large pieces of skin that are going to peel away and detach. Now this can be quite problematic if it spreads into the dermis and deep into the dermis, because the staphylococcus might then spread to capillaries. This is something that's far more common in people that are immunocompromised. So usually in healthy people, Staphylococcus aureus doesn't manage to get that deep into the skin. So S. aureus, very common. About 1/3 of us have permanent populations of S. aureus that live most likely within the nostrils, but perhaps other places on your body as well. And all of us are exposed to S. aureus pretty much every day. It's spread by direct contact, so skin-to-skin contact, or it can be spread by fomites as well. Remember, a fomite is something that's non-living, something that you touch and then someone else touches. So it could be a coffee cup, it could be a towel, something like that. And it's important to note that if you have impetigo, or if your child has impetigo, what you've got in that infection is a fairly large concentrated population of S. aureus that may have a number of virulent factors that aren't seen in most S. aureus. So if it's managed to form that infection, that means that it may have these additional factors that make it more virulent. So if you have a skin infection, you should be careful that you're not spreading it to other people. So, don't touch it. And if you do touch it, don't touch someone else. Staphylococcus aureus is something that can survive off the body. So of course it doesn't have to infect cells to survive. Most of the Staphylococcus aureus that's out there is living on the skin and not damaging the skin, but it can also live for extended periods on floors and doorknobs and telephones, etc. So it is possible, and it's quite easy, to spread it from one person to another through a fomite. And this list here, these things would all be considered fomites. It's a frequent problem for surgical wounds, IV line incisions, catheters. So it's something that has to be treated with a lot of respect in hospital settings. And we talked about nosocomial infections before. These are infections that you pick up in a hospital. So you go into a hospital, you don't have an infection, and you leave with one, which unfortunately is more common than you might think. And about 50% of these infections are caused by S. aureus, just because it is so common and because it's something that it can produce opportunistic infections. Now, about 50% of the infections of S. aureus that people pick up in hospitals are these MRSA strains that we'll talk about in a moment. And the people that are most susceptible, of course, are people that are already ill. A lot of those people in the hospital, no wonder. And also people that are elderly, newborns are quite susceptible, people that are undergoing treatment that will affect their immune system, such as chemotherapy, are also going to be highly susceptible. So how do we diagnose, treat, and prevent infections by S. aureus? Well, for diagnosis, what you would traditionally do is you would take a sample of pus from a wound, and you would gram stain that, and you would look for these gram-positive staphylococci bacteria. So again, remember, staphylo means they grow in clusters, and cocci means that they're spherical. And then once you find those, you could do some additional biochemical tests. We talked about those in the lab in great detail. So we could see if they were salt resistant, we could see if they produce collagenase, etc. There's faster ways to do this kind of thing nowadays, so we can use something called a PCR analysis. And with that, what you do is you use a machine to amplify a specific sequence of DNA that should only be found within the species you're interested in diagnosing. So we would use PCR, polymerase chain reaction, to detect a sequence of DNA that's specific to Staphylococcus aureus. If it's present, then we know we have a Staphylococcus aureus infection, and that's something that can be done in just a few hours. Treatment, so again, traditionally we would use penicillin or a synthetic form of penicillin, and one of the forms that proved to be very effective is methicillin. And methicillin is something that is not proving all that effective against some strains of S. aureus anymore. So we have these methicillin resistant Staphylococcus aureus strains, and that's where the MRSA comes from. And these strains tend to be resistant to most forms of penicillin, and that makes them rather difficult to treat. If we want to prevent the spread of S. aureus, it comes down to just practicing common aseptic techniques. We want to keep things clean. We want to make sure that all our equipment is sterile. We want to make sure that if you're handling a patient, you're doing it with gloves, and then you remove the gloves so you're not transferring Staphylococcus aureus or something else to the next patient. Another genus that can cause problems is Pseudomonas. So Pseudomonas contains several different species of bacteria that are very, very common. They're found in the soil. They're found on your houseplants. They're found on surfaces throughout your house and grocery stores and colleges, etc. In fact, when you went out and did your environmental survey, where you collected bacteria from throughout the college, guaranteed a lot of the colonies on your plates would have been Pseudomonas species. There's one in particular, Pseudomonas aeruginosa, that is a fairly common inhabitant of your skin. And for most people, not an issue, but it's something that can cause opportunistic infections. So let's say you have a pre-existing infection, or you have a deep wound on the skin, or you've got a bad burn that has stripped away the outer layers of the skin, or you've got cystic fibrosis where the mucus within your respiratory system is very, very thick and your body has a hard time clearing it up into the throat. In cases like that, you are prone to an infection with this bacteria. And the most common place to pick up such an infection is in a hospital setting. And you can see there's a big range. 10 to 75% of these nosocomial infections are caused by this species. That range is because it depends on what ward you're in a hospital setting. If you're in a burn ward, this is one that's particularly problematic. It is interesting in that if it's living on your skin in very large concentrations, it gives the skin kind of a greenish tinge to it. So you're seeing some cases here on the left that are fairly severe. We've got lots and lots of this bacteria present. Incidentally, the image on the right shows the bacteria, but that's not their natural coloration. This is a fluorescent microscope picture. So some examples of where Pseudomonas aeruginosa can cause problems. Cystic fibrosis I talked about already. We'll come back to that, but just note that pneumonia is a condition where you have buildup of fluid within the lungs, so within the alveoli of the lungs. The alveoli should be sterile. It's not an area that bacteria are allowed to colonize in a healthy individual. Your system will fight that. But with cystic fibrosis, because there's very, very thick mucus that's produced in that condition, the cilia that move that mucus up into the throat, they just simply can't move it very effectively, and bacteria can get into the alveoli. The bladder, the urinary bladder, should be sterile as well. That's not something that bacteria should be able to colonize. But if you have Pseudomonas on a catheter and it finds its way to the bladder, it can cause an active infection in the bladder. We have gastrointestinal infections as well. These are more common in premature infants and also people with cancer that are undergoing cancer treatments. We have skin infections, and again, most common in bad wounds and in burns. And in some cases, this can cause ear infections as well. Some other skin infections and some rather nasty photos here. Gas gangrene. This is a condition where we've had an infection that is killing cells. It's destroying cells. This creates an anaerobic environment under the skin surface. We have gases that are being produced by the bacteria, and the gases are forming these blister-like structures. So Clostridium is one of the genera that's responsible for this. This is a gram-positive group of bacteria. But again, these are anaerobic bacteria, and these bacteria are feeding on dead tissue, and they're releasing toxins. The toxins are breaking down the connections between cells and killing cells. And then more cells are dying, and this bacteria is feeding on those cells. And one of the byproducts that the cells produces are gases that cause these blister-like structures. Flesh-eating disease. So necrotizing diseases. Necro means dead. So again, we're dealing with bacteria here that are producing toxins that kill tissue and can cause tissue layers to separate. This darkening of the tissue here, that's tissue that's died, so tissue that has become necrotic. There are a whole lot of skin infections that we could talk about. And again, the reason for this is because your skin is a huge target. It's a huge surface, and there's lots of things out there in the world that you come into contact with all the time. I'm not expecting you to memorize all of these, so this next one, for instance, I'm just throwing it out there because it's something that's fairly common that you've probably heard of before. Cat scratch fever, or cat scratch disease, is something that's caused by a bacterium that generally inhabits cats. It's spread from one cat to the next, usually through fleas. Now, occasionally, a cat will have some feces from a flea on its paws, and it may scratch you. And it will introduce that into your skin. This causes pain at the area of the scratch. It causes inflammation at that area. It causes lymph nodes to become swollen in the immediate area, and it may cause fever and headaches and aches and a lot of symptoms that resemble what you'd expect with a flu. So that's if it's actually spreading into your blood flow and becoming a more systemic infection. However, it's usually not harmful. That's generally the case. It can cause some developmental problems in children and so on, but that seems to be very, very rare. All right, on to another skin infection, and now we're looking at viral skin infections, smallpox, caused by variola vera. This was something that historically was a very, very important virus, but it has been wiped out in the wild. There are a few labs, and only a very few labs, that have viable populations of smallpox that they are studying. They're studying it in rats and other mammals. These are containment level 4 labs where people are working in basically spacesuits, and they're taking extreme precautions to make sure that this cannot leave the lab. And you might be wondering, well, if we have successfully wiped this out, why are we permitting this research to go on? Well, of course, smallpox is very virulent. We're always very concerned that new viruses are going to pop up that might have characteristics similar to smallpox. So it is rather important that things like smallpox be studied so that we can understand what makes them dangerous, and we're prepared for that, and we're, hopefully we have some preventative measures in place. But smallpox is an enveloped double-stranded DNA virus. So double-stranded DNA, that's like the DNA that we have. Enveloped means that it has an envelope of lipid around the outside. So it's got a phospholipid bilayer around the outside of it. Smallpox for healthy individuals had a mortality rate of about 30%, which is remarkably high, particularly for something that was quite contagious. It was airborne and spread quite rapidly and quite easily. It's important to note that smallpox and chickenpox are not caused by the same group of viruses, and they are quite different in their effects. Pox is a term for a mark on the skin. So a pox is a pimple-like structure on the skin. And just looking at not only the symptoms, but the way that the pox present themselves, smallpox, you can see that the pox marks were spread all over the body. Chickenpox, it tends to affect mainly the upper body. So even when people were dealing with both of these diseases at the same time, it was fairly easy to distinguish between the two. On to the herpes viruses, and it is a herpes virus that causes chickenpox. So the herpes viruses consist of double-stranded DNA, and that of course is in a protein capsid, and then that is surrounded by a lipid envelope. Herpes simplex virus 1, or HSV1, predominantly causes cold sores. So 90% of people that have this will have cold sores. 10% of the people that have it will develop genital herpes. HSV2 is the opposite. So in 90% of people that have it, it's going to present itself as genital herpes, and in 10%, there's going to be cold sores. Now remember that the herpes viruses can remain dormant. They can go into latency and cause a latent infection because their DNA can be incorporated into your DNA as a provirus. And they do this by inserting the provirus DNA into peripheral nerve cells deep within the skin. And they can just lay dormant there. So for instance, you can have HSV1 and you don't presently display any cold sores, and it can sit there for a long time, and then some sort of stressor can bring about the release of that provirus as its own entity, and it will be decoded to produce proteins and produce an active virus that will then leave that cell, infect other cells, go through the lytic cycle, which is quite damaging. That will destroy or disrupt a lot of cells and cause the actual damage that we see as a cold sore. We also have varicella zoster, which is the virus that causes chickenpox and causes shingles as well. They are the same virus. So with chickenpox, we have the formation of pox structures, which are little pimple-like structures on the skin. They tend to be found mainly on the upper body and face, but they can be more widespread than that. If you have chickenpox later in life, so after childhood, it tends to be a bit more severe, and if it reappears as shingles later in life, that's a more severe condition. Other viral infections that can cause rash-like symptoms would include measles. So we have rubeola, we have rubella. Typically not too harmful, but they can be. They're of significant concern in pregnant mothers because they can be quite damaging to the fetus. Moving on to fungal infections. About 50% of people have yeast growing on their skin, and the most common genus of yeast that we'll encounter is Candida. And Candida is normally kept in check by competition with other microbes, mainly bacteria. So for instance, in the vagina, you have lactic acid bacteria, and as a byproduct of their metabolism, they release lactic acid, and that lowers the pH of the vagina. It makes it more acidic. That's a situation that's not favorable for most yeast, and that will limit the growth of yeast in that area. Now, something happens that destroys the lactic acid bacteria or limits their growth. Let's say, for instance, you're taking an antibiotic to fight off another infection that also kills off the lactic acid bacteria, then the pH of that region will increase. It'll come close to neutral. Fungi will be happier in those conditions, and you may get an explosion of yeast cells. So that would be an example of an opportunistic infection that causes a problem for your body. Thrush is the term that's used when you have an overgrowth of yeast in the mouth. And typically when we're talking about an overgrowth in the vagina, that's vaginitis. Now it depends on who you talk to, what part of the world you're in. Sometimes an overgrowth of yeast in the vagina is known as vaginal thrush. There are other multicellular fungi that can cause problems for the skin, and they tend to produce these ring-like structures on large areas of skin. This is something known as ringworm. That's the name for the condition. Has nothing to do with worms, so don't make that mistake. Ringworm is a ring-like structure formed by hyphae. Hyphae are a bunch of cells all stuck together, so they're strands of cells. And what tends to happen is the growth starts in a localized area, and as the growth moves outwards, it creates this reddened ring-like structure, and that's due to increased blood flow to the active part of the infection. When it comes to fungal infections, quite often they're named after where the infection occurs, and a fungal infection is known as tinea. So tinea pedis is athlete's foot. So tinea pedis, that sounds like a species name. It's not. It actually refers to several different species of fungi that might be growing on the foot and causing this particular condition. So this is an area where biologists and medical researchers kind of disagree with how things should be named. It's not uncommon in medicine to name microbes after the effect they cause, and in the case of tinea pedis, there are several different species that cause that condition that we generally know as athlete's foot. Tinea corporis, this is a condition that occurs elsewhere on the body. And we can also have these tinea conditions occurring on the scalp as well, which is tinea capitis. So recall that myco refers to fungus. If you see myco at the beginning of the word, we're dealing with a fungus. If you're a mycologist, then you're a person that studies fungus. Tinea refers to infections of the skin by fungi, and it might be multicellular fungi or it might be yeast. And the different infections are caused by a number of different opportunistic fungi, and the infections tend to be named by the area where they occur. So tinea pedis, athlete's foot, ped refers to foot. And note that these different infections can be spread by fomites. So if you have athlete's foot, and let's say you're at the gym and you use a towel to wipe your feet and someone else uses that same towel, you may be transferring quite a lot of fungus to them as well. And even though these are fungal species that are out there, they're not found in huge quantities on your skin. So if you wipe your foot and you have athlete's foot, you're transferring a lot to another person. Remember, the incidence of infection has a lot to do with the number of microbes that you're exposed to. So again, it's important to note that fungal infections can be spread by direct contact and they can be spread through fomites. In some cases, there are some fungi that cause infections on pets, like dogs and cats, that can be spread to humans as well. Prevention comes down to keeping the skin dry and relatively clean, so proper hygiene, and avoid sharing personal items such as towels and hairbrushes. Hairbrushes can transfer fungi that live in the scalp, and combs and so on. So you want to avoid sharing items like that if there's any suspicion that you may be carrying these fungi or someone else may be carrying them. To fight fungal infections, there are a number of ointments and creams that you can take, and these typically contain antifungal substances that will attack enzymes that are involved in the production of ergosterol. And ergosterol is a steroid that's found specifically within the membranes of fungal cells. So it's quite similar to cholesterol. Cholesterol has some really important functions in us and in most cells. It maintains the fluidity of the membrane and helps maintain the structure of the membrane. And these ergosterols are a special group of cholesterol-like molecules that are specific to fungi. So if we can interfere with their production, that's going to interfere with the functioning of the membrane. And because we don't have those sterols, this is a target that is not found in our cells, so these creams should not damage our cells. So moving beyond microscopic pathogens, we also have some multicellular parasites. So these are animals that live on the skin or may burrow into the skin. And many of them are only there long enough to feed on your blood. So for instance, we have things like mosquitoes and ticks that will just stay long enough to get a meal of blood and then they leave. They're rather important because of course they can be vectors for other pathogens. We have lice and scabies that stay put for a longer period of time. And incidentally, both of these pictures here are pictures of lice. The one at the bottom there isn't actually scabies, that's a pubic louse. So lice, we have 3 different species that can cause problems for humans. There's head lice, body lice, and also pubic lice. So what you're seeing at the top there, that's a head louse. It will attach to the hairs on your head using the little claws. You can see it's got little pincer-like claws that it uses to attach. I should point out that the first pair of appendages you see on that diagram are antennae, and then there's 3 pairs of appendages beyond that. But they will attach to hairs and then they'll come down to the scalp to feed on blood. Quite often they go to the back of the neck to feed on blood, and they may leave little red welts at the back of your neck or on your scalp. We have body lice, which are unusual in that when they're not feeding, they leave your body. They tend to live in clothing and they'll only attach to the body to feed, and then they retreat back into clothing. And body lice, it's thought they might actually go extinct fairly soon because they're the easiest thing to deal with. Washing clothes frequently is enough to get rid of body lice, and it's something you only generally see on homeless individuals or people that are not able to clean their clothes on a regular basis. And then we have pubic lice. Again, that's what you're seeing at the bottom there. So the bottom left, that is a picture of a pubic louse. They kind of look like crabs, hence the other name for them. And they live in body hair, typically within the groin area, but they can move around. And I think I mentioned before that sometimes they'll end up in men's facial hair. Scabies, I don't have a picture of them here. Look back through your notes, you have pictures of them from before, but scabies are little microscopic arachnids. So they've got 8 legs instead of 6 legs like insects, and they burrow into the skin. They can leave little tunnel-like structures in the skin, and they cause mange in pets, so dogs and cats. And there are some species that can spread from dogs and cats to humans, although those species tend not to be able to exist on humans for a long period of time. They can cause some severe allergic reactions in some cases, and usually what happens is the feces that they deposit in these little tunnels that they make in your skin, you have an allergic reaction to those. So moving on to the urogenital system, which of course consists of the urinary system and the reproductive system combined. When we're dealing with the external genitalia, our main players here in terms of potential pathogens are the same as we've talked about in relation to the skin. So we have the same bacteria and fungi that live in this area as live on the rest of the body. When we're talking about the inner components, the epithelium that lines those organs and those tubes is continuous with the skin surface. So once again, we can have bacteria and yeast and other fungi living on the skin surface, traveling up into those areas. But there are a few players that cause problems within the urogenital system that aren't found on the skin. Now, one thing to note when we're comparing males and females, in females, the urinary system and the reproductive system are completely separate. So of course we have 2 separate openings involved there. But when we're dealing with males, the systems are combined. So the urethra is going to conduct semen, and it's also going to conduct urine. The other thing to note is that the length of the urethra in males is quite a bit longer than it is in females, and that has to do with how the 2 systems are combined in males, and also the presence of the penis that lengthens the urethra. So it's a rather difficult trip for bacteria to get from the opening of the urethra at the end of the penis to the bladder. It's a far shorter trip in females, and that's why females tend to have a lot more bladder infections than do males. But if you look at the list of potential pathogens in both these systems, you'll notice that there is a lot of overlap between the 2. So let's start with infections of the urinary system. UTIs are urinary tract infections, and there's 2 broad types based on the region that's infected. So we have lower UTIs. These involve the urethra, which is the tube that carries urine from the bladder to the outside of the body, and the bladder. These are fairly common, again, more common in females than in males. The bladder has very smooth, tough walls to it, as does the urethra. So pathogens do have a hard time staying put within this region. Then we have upper urinary tract infections, and this would include the ureters and the kidneys. So the ureters are the tubes that carry urine from the kidneys to the bladder. These are less common and also far more dangerous. So if pathogens, fungi or more commonly bacteria, manage to get to the kidneys, they may spread into the blood flow and cause a potentially very damaging systemic infection. Diseases can also be spread by sex, so sexual activity. And there's 2 terms here that are sometimes used interchangeably, although they do have different meanings. First of all, we have a sexually transmitted infection, or STI. This is the condition whereby you have a pathogen living in the urogenital system that was transmitted by sexual intercourse or sexual behavior of some sort. You can have an infection without having any symptoms. So for instance, it's possible to have a herpes infection that is in latency. It's possible to have a chlamydia infection where you have live chlamydia bacterial cells reproducing within your urogenital tract, but they're not producing any symptoms. An STD, a sexually transmitted disease, is where you have symptoms resulting from that. So let's say you have chlamydia, but you're not showing any symptoms. You have an STI. Now let's say you have chlamydia and you are showing symptoms as well. You have an STI and you also have an STD. The most common cause of urinary tract infections is E. coli. And E. coli, of course, is beneficial when it's in the intestine, except for a few strains that can cause problems. But for the most part, E. coli is something that's beneficial. It helps us break down our food a little bit further in the colon, and it produces some vitamins that it passes on to us. But we're talking about E. coli that are somewhere they shouldn't be. In terms of STIs or STDs, chlamydia is the most prevalent in Canada. And we'll come back to this and talk about this in a bit more detail. And gonorrhea is the second most common STI or STD in Canada. Syphilis, interestingly, is also on the rise. This is a condition that's caused by a spirochete, a spiral bacterium that has been historically very important. And we are actually seeing more cases of it recently. The majority of E. coli in the intestine are not capable of causing bladder infections. However, there will be a few individuals, so we'll have populations of E. coli in your intestine that have some additional virulence factors that may make them problematic if they find their way to the bladder. Among these things, we have what are called P. pili or P. fimbriae. These are fimbriae, which are strands that stick out from the bacterium and allow the bacterium to stick to epithelial cells. And this specific form of these pili or fimbriae allows them to stick to the interior of the bladder. But again, most bacterial cells, most E. coli cells are unable to do this. Let's look at the trend of urinary tract infections with age. And again, we're dealing here with E. coli predominantly. If we look at both males and females as infants, there's a fairly high incidence of urinary tract infections. That makes sense. Infants are sitting in diapers. If the diapers are not changed frequently, they may be sitting in their own feces. And of course, feces contain E. coli. And we see lots of urinary tract infections in infants for that reason. Now, throughout most of adulthood, we have urinary tract infections being relatively common in females, but not common at all in males. And again, the reason for this is the E. coli is coming from the anus, and it has to make its way to the bladder. And in females, that's a fairly short trip. And if we've got poor hygiene contributing to this, it's something that can happen fairly frequently. In males, there's a larger distance between the urethra and the rectum, and also the urethra itself is quite a bit longer. So it's a long trip for E. coli to get from the opening of the urethra to the bladder. Of course, every time you urinate, you've got a fairly powerful stream of urine that flushes out that slippery tube. Now, if we look at older individuals, we can see that in old age, in the 70s and 80s, we have, again, a lot of cases in males. And the reason for this goes back to the same reason we have more urinary tract infections in infants. Basically, with elderly individuals, they may also be wearing adult diapers. They may also not be able to clean themselves effectively, and hygiene becomes a big issue. If you have a urinary tract infection with E. coli, it can range from rather mild to very severe. If you've got a lower UTI, then that's not a severe condition. It can be treated fairly easily, and it should be treated right away because we don't want to wait until it develops into an upper urinary tract infection. That would be a bad thing. So with an upper urinary tract infection, there can be severe abdominal pain and fever. And of course, once the bacteria gets into the kidney is very well vascularized. So the bacteria may make its way into the blood, and then we've got a very dangerous systemic infection. With urinary tract infections, there tends to be cloudy urine, and that's because we've got leukocytes and debris in the urine, and it may be possible to take some of that urine, spread it on a petri plate, and culture the pathogen. So prevention of E. coli infections of the urinary tract, first of all, careful hygiene is very important. So again, especially in females, you want to make sure that feces don't end up at the opening of the urethra. Sexual practices as well. So anal sex followed by vaginal sex is a bad idea for a few reasons, most predominantly E. coli infections. Treatment, so E. coli quite often is resistant to penicillin, and that's because penicillin has been around a long time. We all have E. coli in our systems, and there are many strains of E. coli that are resistant. But there are other antibiotics that can be used, and quite often antibiotics are used as a cocktail, so more than one antibiotic might be given. Now the problem with using antibiotics is we have to be aware that we might also kill lactobacilli in the vagina, and that's going to create a near neutral condition by getting rid of those acid producing bacteria, and then we can have subsequent problems with yeast. Chlamydia is often asymptomatic. So you have the infection, you have the STI, but you don't have the symptoms, you don't have the STD. That's something to keep in mind. So you might have it and not know. You might have a partner that has it and they don't know. So of course, wearing a condom will help protect you against chlamydia and several other STIs that don't result in symptoms. Another thing that's interesting about chlamydia is it can infect epithelial cells outside of the urogenital system. Now, it can't survive very long in those epithelial cells. It'll survive for years and sometimes even decades in epithelial cells of the urogenital system, but it can survive, for instance, in the throat, in the epithelial cells lining the throat for a short period of time. So it's possible that you could have oral sex with someone who has chlamydia. You could pick up the chlamydia and it could reproduce within your throat. And typically what happens is it causes inflammation of the throat. It becomes quite red, it becomes sore, and it produces symptoms that look a lot like strep throat. If you have that and you have oral sex with someone else, you can actually give them full-blown chlamydia through the act of oral sex. So chlamydia is kind of fascinating in that although it's a sexually transmitted disease, it's not always transmitted from genital to genital. It can be transferred genital to the oral cavity and then back to genitals. So chlamydia is, as I mentioned, asymptomatic in a lot of people, especially in women. So 75% of women will not show any symptoms. If you have symptoms, that consists of a vaginal or penile discharge. So you have kind of a whitish sort of thick, viscous discharge, and also a burning sensation while urinating. And once again, chlamydia cells are viral-like in some ways. They do have a metabolism, but it's simplified. They can't produce their own ATP, so they have to steal ATP from cells. And that means they're only active when they're inside cells, a lot like viral cells. They don't survive very long outside of cells. So what you're seeing here in these diagrams is chlamydia cells living inside epithelial cells. So the one on the bottom left there, for instance, the red is the result of a fluorescent dye that is bound to chlamydia cells that are living inside epithelial cells that are being depicted with a green fluorescent protein. If you have chlamydia and it's left untreated, it can exist for years in your body, and it can cause a lot of rather serious problems. So in women, it can eventually spread into the uterus and into the fallopian tubes or oviducts and cause infertility or ectopic pregnancy. Ectopic pregnancy is where the embryo implants somewhere other than the uterus, so it implants in the fallopian tubes, and that can be rather serious. There's not enough room for an embryo to develop in the fallopian tubes. In men, chlamydia can sometimes spread to the testes, and if it does that, it will cause damage and cause infertility. Oddly, in some cases, chlamydia infections, if left untreated for a very long period of time, can lead to some types of arthritis. So there are many types of arthritis where the immune system attacks connective tissue, and this sort of arthritis can be prompted by chlamydia infections. Traditionally, if you have a bacterial infection of some sort, what you would do is you would take some body fluid that may contain that bacteria and you would culture it. You can't do that very easily with chlamydia. You can't just take a sample that has chlamydia, spread it onto a petri plate, and have the bacteria grow so that you can look at them under a microscope, because of course chlamydia has to live inside other cells. It won't live on a petri plate. So in the past, if they wanted to culture it, it was quite tricky. They had to have a tissue culture, and then they had to grow up the cells in that tissue culture. Nowadays, though, we can detect chlamydia very quickly, and we use PCR. I've mentioned PCR a few times now. PCR, again, is just an automated process that can amplify or make copies of very specific sequences of DNA. So if you suspect you might have chlamydia and you go in for testing, what they'll do is they'll take a small amount of urine, and then they will run that urine through this PCR machine, and what it will do is it will make copies of a piece of DNA that is specific to chlamydia. So if after this test occurs that DNA is present, that is a positive test for the presence of chlamydia. Treatment is macrolide antibiotics, and typically this will be a pill that you take, and you may have to take it twice or more, but it's a very simple treatment. Macrolides are antibiotics that will shut down protein production. And if you remember, we talked about these a while back. These are antibiotics that will specifically attack prokaryotic ribosomes. So they shouldn't harm your ribosomes, but they will shut down protein production in chlamydia. Gonorrhea is caused by another bacterium, and it produces symptoms that are quite similar to chlamydia. They can be difficult to tell apart, and like chlamydia, it can very often be asymptomatic, so you may not know that you have this STI. If you don't know that you have it, you may be spreading it to other people. You want to make sure that this is something you deal with. If you don't deal with it, it can cause problems later in life. It can lead to sterility in both males and females. So like with chlamydia, the symptoms are discharge and painful urination. In females, we may also have bleeding, and sometimes cramps may develop as well. Gonorrhea can be treated with penicillin and synthetic penicillins. Unfortunately, there are quite a few forms that are now resistant to penicillin and its derivatives. It's interesting, when penicillin was first invented and became really popular during the Second World War, it was used to treat battlefield wounds, amputations, that kind of thing, but it also proved very, very important for soldiers that were getting the clap or getting gonorrhea from visiting prostitutes during the war. Syphilis. Syphilis is a rather old disease. It's been with us for quite a while. It's caused by a spirochete bacterium. Very important historically, because if left untreated, it causes disfiguration. It can cause changes in bone structure and so on. And there are a lot of important individuals in human history that we know from drawings and paintings, probably had syphilis. It's an interesting pathogen in that the symptoms produced by this mimic a lot of other diseases, so it can be confused with a lot of other things. So the primary stage, when you first might notice it, usually there are genital sores or cankers. So those are like the canker sores that you might have in your mouth. That's something quite different, typically, although this may produce oral cankers as well. After that, you may have rashes on your hands and feet, usually on the bottom of the feet and the palms of the hand. You can get a fever, sore throat, aches and pains and hair loss and a whole bunch of other things. And then if this is ongoing and not treated, it can be very damaging. So it can move into muscle, into bone, into the central nervous system. So it can cause systemic organ damage. It can cause paralysis in some cases, dementia, and also disfigurement as well. So it can affect the maintenance of bone. It can affect the facial bones and change the shape of your face, for instance. Another thing that's rather concerning is that it can be transmitted across the placenta to the fetus. And it wasn't uncommon in the past that people would be born with syphilis, and then they'd have a lifetime dealing with syphilis, and as you can imagine, that proved to be quite disfiguring. Moving on to viral infections of the urogenital system. We've got genital herpes. Remember, that can be caused by HSV1 more commonly, but also by HSV2. We've got genital warts that are caused by HPV. We talked about this when we talked about viruses. Remember, this is a non-enveloped, circular, double-stranded DNA virus. And we do have vaccines for this. It's very common. So a very common STI, estimated that perhaps as many as half of all sexually active people will get this at some point in their life. It's something that definitely needs to be addressed because it can greatly increase your risk of certain types of cancer, especially cervical cancer. So remember that what viruses do is they can embed their genetic material into yours. They can integrate their genetic material into yours. And if they do that, they do it somewhat indiscriminately, and they may interrupt genes that are involved in controlling the cell cycle. And if that's the case, and they turn off genes that normally tell cells to stop dividing, then that may lead a cell to divide uncontrollably and become a cancerous cell. When it comes to fungal infections of the urogenital system, we have one genus that is responsible for the vast majority of them. It's Candida. So Candida is, once again, a yeast. It's a single-celled fungus that's fairly common on the body, and it's not uncommon for it to be present in small amounts in the vagina, but it's held at bay by the pH in the vagina, which should be a bit acidic. The acidity is coming mainly from waste products that are released by lactobacilli. And if you kill off the lactobacilli, all of a sudden the yeast can take off. But also I should mention that if you kill off the lactobacilli, then any E. coli that have found their way to the vagina have less competition as well. So losing those helpful bacteria, those lactobacilli, is opening up the door for a lot of potential opportunistic pathogens. And finally, we do have a few eukaryotes that can cause problems within the urogenital system. The most common one is Trichomona. Trichomona vaginalis. As the name suggests, it lives in the vagina. This is a protist. It's a single-celled eukaryote. It doesn't have any mitochondria, which is unusual for a eukaryote. Because it doesn't have mitochondria, it's anaerobic. It's actually poisoned by oxygen. And there's not a whole lot of oxygen in the vagina. It creates a rather fishy odor. It's something that is not uncommon in females, but very uncommon in males. Now, it's possible that males may transmit it from one female to another, but it doesn't seem to affect males in any way, and it doesn't seem to be long-lived on males. Incidentally, the same is true for yeast. Males can have yeast living on the skin, so they can have, a few yeast living on the penis, the skin of the penis. It's possible that males can transfer yeast from one female to another, but typically there's more yeast within the vagina than there would be on the penis. So both of those are things that generally don't cause problems for males. And then we've also talked about pubic lice, and that's a pubic louse there, so it does look quite crab-like. As mentioned, they can live on any body hair, but generally they prefer the groin. So moving on to the respiratory system. It's convenient to divide this into two parts. We have the upper respiratory system, which is everything above the epiglottis. The epiglottis is the little flap that folds over and prevents food from going into the trachea and instead redirects it into the esophagus. And then we have the lower respiratory system, which is below the epiglottis. The upper respiratory tract is going to include the nasal cavities, the upper part of the pharynx, so the nasopharynx, which is the connection between the nasal cavities and the throat, and the oropharynx, which is the part of the pharynx that's right behind the oral cavity. And it's also going to include the sinuses and the part of the ear as well, because remember, the middle ear is connected to the pharynx by the eustachian tube, also called the auditory tube. The lower respiratory tract is going to be the trachea and also the branches that come off the trachea. So the bronchi, the bronchioles, and finally the little sacs at the end, which are the alveoli. And we have a lot of potential pathogens that could find their way here. Some of them are unlikely to do so in healthy individuals, but reasonably likely to do so in immunocompromised people. So for instance, fungi, there are some rare fungal infections that can occur in healthy people, but usually fungal infections of the lung occur because there's a pre-existing infection or because there is an immunodeficiency of some sort. Okay, so starting in the throat, strep throat caused by streptococcal pharyngitis. Remember that strepto means that the cells grow in chains, as you can see in the diagram here. Coccus refers to the fact that they are spherical. If you grow these on blood agar, you see a clearing around the blood agar indicating that they can break down red blood cells and presumably break down other cells as well. And they can damage epithelial cells. So if you have this infection, the epithelial cells are damaged, there's increased blood flow to the area, and that causes redness and pain and swelling. There's a number of different Streptococcus bacteria that can cause infections within your body. They're named usually after their characteristics. They're named after proteins that they have exposed on the surface that function as virulent factors. So these proteins are used to attach to cells and damage cells and so on. Some Streptococci species may also have a capsule around them, and as we've talked about before, if a bacterial cell has a capsule around it, that makes it far more difficult for your immune system to deal with. It makes it difficult for white blood cells to grab onto them, engulf them, and break them down. Sinusitis is an inflammation of the sinuses. So again, itis on the end of something means an inflammation of. There are a few different bacteria species that can cause this. We won't get into all the different types, but notice that S. aureus is on here. And incidentally, there are some bacteria that have influenzae as part of their name. Don't get that confused with the influenza virus. That's something different. We have otitis. Otis refers to the ear. So this is an infection of the inner ear, which can be rather serious. It's not too uncommon for infections of the inner ear to spread and become systemic infections in some cases, or to damage the inner ear and cause permanent damage. We have pertussis, which is whooping cough. The lower respiratory system is protected by what's called the mucociliary transport system, also known as the ciliary escalator. So you have pseudostratified ciliated epithelial cells. They've got cilia at the top on their apical end, and we've got these goblet cells that produce mucus, and the mucus will sit on top of the cilia. Any debris that you breathe in, like dust for instance, any spores that you breathe in, any active bacteria that you breathe in, should end up stuck to this mucus, and then that mucus is going to be wafted up into your throat by the constant beating of the cilia, and then you swallow that mucus along with anything it trapped. That goes down into the stomach, where stomach acid and enzymes should destroy anything that you have caught. Now, if you have a pre-existing infection, so let's say, for instance, you have influenza, and the influenza virus is attacking those ciliated cells, they might not be working as well as they could, and that's going to make you more susceptible to other infections, because now that ciliary escalator is not working at full efficiency. If you're a smoker, smoking actually damages these ciliated cells quite a bit, and you have a less active ciliary escalator. So there are a number of conditions that can damage this system and make you more susceptible. Now, what this ciliary system does, like I said, it traps materials, it brings it up into the throat so you can get rid of that. It greatly limits the amount of material that can get down into the lung itself. The ciliary system doesn't continue into the bronchioles though, and into the alveoli. Instead, in the alveoli, you have macrophages, so large white blood cells that wander around on the surface of the alveoli, so the interior surface, and gobble up anything that shouldn't be there. So ideally speaking, in a healthy person, the interior of the lung, the alveoli of the lung, should be completely sterile. It's also worth noting that you have this transport system in your nasal cavities as well. So if you are sick and your nose is running, what's happening is the cilia are not beating at the rate they should be, and that's because you've got maybe a viral infection, for instance, that's doing some damage to those ciliated cells. The nose is producing more mucus than usual, and it's not moving it back to the throat very effectively, and that's causing excess mucus to flow out of the nose. Also, if it's really cold and your nose starts running, what's happening is the cold air in the nose is slowing down the metabolic rate of those cells. The cilia aren't moving, they're not as active, and some of that excess mucus and fluid is leaking out of your nose. Also, it's worth noticing that if you're crying, your nose runs. And the reason for that is the fluid that runs over the eye and helps remove bacteria and debris from the eye ultimately drains into the nose. And if your eyes are watering because you're crying, that means that your nose will water as well because it can't move that fluid fast enough back into the throat. So again, this system is going to move stuff from the nose towards the back of the throat so that it can be swallowed, and this system will move stuff from the trachea up to the throat so it can be swallowed. Tuberculosis can be a very long-term chronic infection. It's caused by a mycobacterium, Mycobacterium tuberculosis, well named. And if you recall from when we talked about staining techniques, this is a bacterium that has a layer of mycolic acid. So it's got this hydrophobic, kind of waxy layer that makes up part of the cell wall, and that helps protect it from your immune system, specifically from your white blood cells. These cells divide very slowly. They're very slow growing. This wall is rather complex, and it takes a while to build all the materials that go into the wall. So they don't divide nearly as quickly as other bacteria. As an example, E. coli can divide every 20 minutes. Tuberculosis takes a day or more to divide once. They're acid-fast bacteria, so remember acid-fast staining, that allows us to identify cells with this hydrophobic, rather tough mycolic acid layer. This is a really important disease. It's been important historically, and it continues to be very important in many parts of the world. It's a major killer in much of the world. And in fact, historically, it has sometimes been termed the White Death or the White Plague. Remember, we've got the Black Death or Black Plague that refers to bubonic plague. Tuberculosis, also quite serious. So the tuberculosis bacteria can live inside alveoli. Again, it's very slow growing. It divides very infrequently, but it's also very well protected against your immune system. White blood cells will come along to the site of infection. They'll leave the capillary and enter into the alveoli and become macrophages, these large phagocytic cells. And they will gobble up the bacteria, but they can't destroy them. And in fact, the bacteria can divide inside the white blood cells. So your immune system keeps throwing more and more white blood cells at these bacteria. They can't outright destroy the bacteria, but they can kind of wall off the infection. And what happens is we end up with something called a tubercle forming. And you can see in the bottom diagram here, what we've got is this inner lumen, this space in the middle, and that's where all the white blood cells have been destroyed. And the bacteria have been released and they're very slowly dividing. New white blood cells are coming in, they're engulfing the bacteria, they're slowly being killed. But we have this shell of macrophages that have walled off this area of infection. And they're slowing the spread of the bacterium and they're preventing it from getting to the rest of the lung. So we can have lots and lots of little pockets of infection where we had perhaps just one cell settling out and dividing very slowly, but that infection is contained within this shell of white blood cells. These tubercles can eventually burst, and if they break open and spill their contents into a bronchiole, then all of a sudden you've become infectious in that you can transmit this to another person. So now when you cough, you're going to be releasing active cells, active bacterial cells that someone else may breathe in. So long as the cells are all walled off in these tubercles, you're not going to spread the disease. And you can go through periods of latency. If the cells are all walled off, you won't have the symptoms. You won't be coughing. You won't be spreading the disease in this case. But when those tubercles rupture, they cause lots of damage, not only to alveoli, but to the bronchioles as well. And you may be coughing up blood, but you're also coughing up active bacteria. There are several means of detecting tuberculosis, and how effective these are going to be depends on whether the patient has an active infection or if they have a latent infection. If they have an active infection and these tubercles are rupturing and releasing bacteria, then we can find the bacteria in their sputum. The sputum is what they cough up. So we have the person cough up some material, we put that on a slide, and we acid-fast stain it to see if we have acid-fast bacteria. We can do the same thing on a slide, but this time using immunofluorescence, and that's where we have antibodies that are bound to a dye, usually a fluorescent dye, that will recognize antigens on the bacterium if it's present. We can do PCR, and this is where we're looking to see if there's a specific sequence of DNA present that is only found in the tuberculosis bacterium. And we can do ELISA. These are other forms of assays or tests that use antibodies. So the skin test, this is an example of a test using antigens to test for the presence of antibodies. What you do is you take a solution that contains antigens from the tuberculosis bacterium, so these would be proteins, glycoproteins, on the surface of the bacterium. You inject them into the skin and you form a little pocket of fluid under several layers of skin. And if this person has been exposed to tuberculosis at some point in the past, they will have produced antibodies against those antigens. And then we're going to have a reaction forming that produces this swollen area. We can use chest x-rays as well, so we can look for those tubercles. And if it's a bad infection, we may have a lot of tubercles. The tubercles may have ruptured as well and basically melded together to form these light areas on the x-ray. Remember that on an x-ray, the dark areas are areas where there's nothing to impede the x-ray. So your lungs contain a lot of air. The x-ray should pass through the lungs. The lungs should be quite dark or even black. on an x-ray of a healthy person. If you see a lot of white material, that would be tubercles in this case. So that's what you're seeing where the red arrow is. And of course, the big white spot in the middle is the heart. So let's compare latent TB infections and active TB infections, or in this case, disease, because it's causing symptoms. So with a latent TB infection, there aren't any symptoms and the person won't feel sick. If they have an active TB infection, they're going to have a very bad cough and a lot of pain associated with that. They're going to be coughing up a lot of sputum, and it will likely contain some blood because a lot of tissue is being destroyed and ruptured. They're going to be very weak and fatigued, lots of weight loss and chills and fever. And this can be, like I said, a really nasty disease. In terms of transmission, if you have a latent TB infection, you're not going to spread it to other people because the bacteria have all been walled off. They're not being released in your breath. The infection is going to be kept in check by the immune system. It can't destroy it completely, and you can have this latent infection for years or decades, but your immune system is keeping it in check. But once you develop the active TB disease, then the bacteria is going to be found in the respiratory droplets that you're coughing out. In terms of diagnosis, if you have a latent TB infection, you're going to have a positive skin test, usually, but not always. If you have a fairly mild infection, as in a rather small number of bacteria, and you've been pretty successful at walling it off, you might not have a lot of antibodies in your system. If you have the active TB disease, you're going to have a positive skin test almost always. Now, the skin test is not always 100%. You can get false positives sometimes. And also keep in mind that if you've been treated for TB, but you had it in the past, you'll still have those antibodies. So even if you don't have TB, but you have been exposed to it in the past, you will probably test positive. For chest x-rays, you'll likely have a normal chest x-ray if it's a latent infection, especially if it's early on. With an active TB disease, you're going to have an abnormal chest x-ray because these tubercles will have merged together and collapsed, and you'll have some fluid buildup as well, and that will be seen as an x-ray absorbing spot because of this fluid. With a latent infection, you'll have a normal sputum test. You won't have any bacteria in it. With active TB, you will likely have bacteria present that you can test for or that you can culture. And the treatment, if you have active TB, they will give you a multi-drug treatment. One of the drugs is probably going to be isoniazid, and that is a drug that prohibits mycobacteria from making mycolic acid. So it attacks the enzymes that are involved in that. So that will hopefully help weaken the cell wall, but that's not enough. There will be other drugs as well that will attack different parts of the bacterial metabolism. So it could be a number of different drugs that are used, a cocktail of drugs. If you have a latent TB infection, sometimes just using the isoniazid is enough because that will slow the growth rate of the bacteria even further, to the point where perhaps your immune system can actually do something about it. This is an ongoing drug therapy though. The bacteria are slow to divide and grow, and it's also a pretty slow process to kill them and get rid of them. Pneumonia. Now, pneumonia refers to fluid in the lungs. That's really all it means, and there's a number of different things that can cause a buildup of fluid in the lungs, but usually it's a bacterial infection. Quite serious, very, very common in hospitals. People that go into a hospital that have an immune disorder or another infection of some sort that is really tapping out their immune system may develop an infection of the respiratory system, and that will quite commonly lead to pneumonia. Now, if you have fluid building up in the alveoli, as you can imagine, they're going to be quite ineffective at taking in oxygen. So this is going to have a huge impact on oxygen and carbon dioxide exchange at the lungs. So here you've got a case of someone with pneumonia, and again, the white spots there are due to buildup of fluid. So we have alveoli filling with fluid, and that's creating these lighter colored areas. Lots of different bacteria that can cause this, and note that S. aureus is on there. Again, Pseudomonas, even E. coli, a whole bunch of different types of bacteria can be responsible for pneumonia. And some viruses as well. Viral infections of the respiratory system. So we have the common cold and we have influenza. Those are the big ones. The common cold is caused by a number of different viruses. There's at least 200 different types of viruses that cause a set of symptoms that we recognize as being a cold. Colds are highly contagious, and most of these viruses, though, don't produce severe symptoms. So they have high pathogenicity, but low virility. And it's rhinoviruses and coronaviruses that are predominantly responsible for colds. They're spread by respiratory droplets. So again, these are tiny little droplets or aerosols that you produce when you sneeze or cough. Remember that you should not be using antibiotics for viral infections. Antibiotics are effective against bacteria. They don't do anything with viruses. And part of the reason we have this problem with antibiotic resistant bacteria is because in the past, doctors and other medical professionals didn't think anything about giving antibiotics out if anyone was sick. They figured, well, not sure if it's a virus or a bacteria, but if it is a virus, it won't do any harm. But of course, what happens is it will select for resistant strains. So antibiotics should be used carefully and only when you're pretty sure that you're dealing with a bacterial infection. Now, there are some more severe members of these groups. SARS, I talked about before. The best known outbreak of SARS was in 2003, and there were about 8,000 people that were affected, and I want to say 770 or so that died as a result. SARS has a low pathogenicity in that it doesn't spread very readily, not nearly as readily as the common cold, which is highly contagious and thus has a high pathogenicity. But SARS is more virulent, meaning that if you do get it, it's more damaging. And of course, with COVID-19 today, this is a SARS-related virus. It's in that same group of viruses. It is not as deadly, not as virulent as SARS, but it is more contagious. So of course that's the fear with it. I've talked about that before, and I won't spend a lot of time on COVID-19 because I'm hoping you guys are all experts on it now. But with COVID-19, it's somewhere in the middle between a common cold and SARS in that it's much more dangerous than the common cold, but it's not nearly as contagious. Like the coronaviruses, the influenza virus is also enveloped. So it has an envelope made of lipid, and this is a bilipid layer that it is stolen from a cell as it left the cell by budding. In the influenza viruses, we have proteins embedded within that membrane that allow the virus to enter a cell and proteins that allow the virus to leave a cell. So the hemagglutinin, the H proteins, are used by the virus to attach to a host cell and enter the host cell. The neuraminidase group of proteins are proteins that are used to leave the cell. So when we're identifying different groups of influenza, we refer to the type of protein that is present. So there's 9 different N proteins that have been identified and 16 different H proteins that have been identified. So for instance, H1N1 tells us that we have the first type of the H protein and the first type of the N protein. H1N1 influenza viruses caused the Spanish flu of 1918 and also a swine flu outbreak in 2009. The reservoirs for influenza are humans, of course, but also birds. So we have bird flus and swine, swine referring to pigs. So with influenza, the symptoms are short-lived, but they can be rather severe. And influenza is a disease that kills a lot of people. Not that many healthy people. Generally, it kills people that are elderly or that have other coexisting issues, such as compromised immune systems. But as you know, if you've had a flu, it doesn't last very long, but it really kind of kicks your ass more than a cold would. So we've got cough and chest discomfort that are caused directly by the virus attacking the epithelial cells within the respiratory system. And then the immune response brings about fever and chills and headache and muscle aches. Those are all part of the immune response. So remember that fever is a full body response that makes things more difficult for the pathogen and also makes the immune system more effective. The reason you get chills is what happens in your body is the hypothalamus, part of your brain that acts as the thermostat, tells your body that it's cold. And that causes the body to raise its temperature. So it causes muscles to quiver and release excess heat, for instance. But basically, even though you might be 37 degrees, your hypothalamus, in order to produce the fever, tells your body that it's not warm enough. And then your body goes through all these processes to raise the temperature. But until it gets up to that elevated temperature, your body is being told that it's cold. And that's what's happening to create these chills. So the virus is going to kill ciliated epithelial cells, and that can be a problem because ciliated epithelial cells, of course, are part of that mucociliary elevator. And if we have damage to a lot of those cells, then that makes us susceptible to other infections. So often we can get a secondary bacterial pneumonia infection. So you have a really bad flu, you're no longer able to get that mucus up into your throat and swallow it, and some bacteria that you breathe in actually manage to get down to the alveoli and they cause this secondary infection. Influenza viruses undergo rapid change, specifically with respect to the proteins that are exposed on the surface. And this can happen through one of two ways: antigenic drift or antigenic shift. So remember that all viruses are going to carry with them genetic material, and that genetic material is going to be copied by the host cell, and it's going to be used to make messenger RNA and ultimately protein by the host cell. Viruses can carry some of their own enzymes with them that the host cell will use in this process. The copying of genetic material in viruses tends to be somewhat inaccurate, and lots of errors get introduced, and this brings about mutations. We have random mutations that are slow and steady. They're occurring all the time, and a lot of them are going to make the virus ineffective. But remember, we're dealing with hundreds of millions or billions of viral particles when we have a virus infection. Some of the changes might help the virus. It might help them spread and infect other cells, for instance. So antigenic drift is just the slow change in the genetic makeup and the surface proteins due to random mutations. Antigenic shift is when we have two similar influenza viruses that will come together. So here we've got an example where we've got a human strain of influenza. It's something that only infects humans. And we've got an avian strain that only infects birds. Avian means bird. The avian strain is highly pathogenic, which means it spreads quite easily. Now imagine if somehow we have a slight change in either one of these two viruses so that they can now live in the same host. If they can infect the same host, then it's possible that we could get swapping of proteins and genetic material between these two strains, and we can suddenly come up with something that's different. So now we've got a case here where you can see we've got a new form of the virus that has obtained some of the H proteins that made the avian strain very pathogenic. So you can see we've incorporated genetic material from two different viruses, and we've made a brand new virus. So this is antigenic shift. Doesn't happen very often, but it's very fast. So it's something that will happen in one host. We can't really predict this, not that we can predict drift all that well either, but antigenic shift is something that can happen all of a sudden in a small population of birds or pigs or humans and give rise to something quite new all of a sudden. So as an example here, we have an avian bird reservoir for this H5N1, and we have a human form of influenza. You can see that down the bottom there, that only affects humans. Now imagine if that avian form and that human form mutate slightly so that both of them can coexist in a pig, in swine. If they both coexist in that host, then we can get this exchange of bits and pieces of the virus. So you can imagine, let's say within the pig, we have that human virus and we have the avian virus, and they both infect the same cell. Now the proteins that are coded for by the genetic material and the genetic material itself is going to get reshuffled in that host cell. And some of the virus particles that leave are going to be a recombination of those two different viral strains. And now we have something brand new that perhaps has become more pathogenic, or maybe it's become more virulent. Moving on to the digestive system. We can divide the digestive system into two broad parts. We have the upper digestive tract, which is the mouth and pharynx, and esophagus and stomach. Also included in the upper tract would be the accessory organs, the liver, the gallbladder, and the pancreas. Then we have the lower digestive tract, which is the small intestine and large intestine, or colon. So let's start in the mouth, or oral cavity. We have a number of different species of bacteria that are found in the mouth, and they can lead to dental caries, also known as tooth decay or cavities. And typically these bacteria will live in rather thick biofilms on the surface of the teeth and just below the gum line. That's plaque. These bacteria can also cause an inflammation of the gingiva, which is the gums. So looking at those biofilms, they can be quite thick, and they can contain as many as 400 different species of bacteria, and many of these species are implicated in causing tooth decay and gingivitis. The stomach is incredibly acidic, and that makes it a barrier to most bacteria. So most vegetative, active bacteria that might be in your food aren't going to survive the trip through the stomach. Now some endospores can, and also some cysts of protists can survive that trip as well. Generally though, we don't have active bacteria living in the stomach, with one very notable exception. So Helicobacter pylori is a gram-negative bacterium that can survive the harsh conditions of the stomach. It's microaerophilic. What that means is it needs just small amounts of oxygen to survive. And there is a very small amount of oxygen in the stomach. A lot of people have this bacterium and it doesn't cause any problems, but it can cause ulcers. You can have ulcers forming in the stomach, you can have ulcers forming in the duodenum, as you're seeing here, and ulcers are lesions within the epithelial lining. You can have them in the mouth as well. That's what we call canker sores. Those are not caused by the same bacterium though. Now these are rather painful if they're in the stomach, because the underlying areas will be exposed to these harsh acidic conditions. The inside of the stomach is normally coated in a very thick layer of mucus, and that helps protect the lining of the stomach from damage by the acids that are generated within the stomach. We also have thick layers of mucus within the rest of the digestive system that help prevent bacteria from getting to the epithelium and damaging those tissues. Cholera is hugely problematic in some parts of the world. It's a historically important disease. It's been around for quite a long time, and it's something you get from contaminated water. So in areas where there's poor sanitation and poor treatment of water, cholera is still quite a problem. And what cholera does is it causes your body to lose a lot of water very quickly. It can cause vomiting as well, but mainly this is through diarrhea. The cholera bacteria produces an AB enterotoxin. I talked about those in the last topic. And what this does is it interferes with the movement of sodium and chloride and some other ions across the cell membrane of the epithelial cells that line the small intestine. Basically, it prevents sodium from being taken up from the lumen of the intestine into the cells, and it causes the cells to dump chloride into the lumen of the intestine. So what that means is the lumen, the space inside the intestine, fills up with a lot of salt, and that draws water out of the epithelium of the intestine. So instead of producing solid, drier feces, you produce a lot of fluid and you lose that as quite voluminous diarrhea. So people throw around the term food poisoning quite often. It's something that's not used correctly in a lot of cases. Poisoning means consuming a toxin. So you're consuming a chemical, a compound that is toxic, and the effects you're experiencing are due to that toxin. If, let's say, you eat something bad and you pick up salmonella and you have an active infection of salmonella, that's technically not poisoning, that is infection. So a food infection is an illness you get from consuming an active microbe. The microbe is active in your intestines and it's damaging the lining of the intestines or disrupting the functioning of the digestive system. So again, salmonella, bacteria species, it's the active living bacteria that causes the problem. Technically, this would be an infection and not a case of poisoning. In poisoning or intoxication, again, it's a non-living toxin that's causing the problem. So there's toxin in the food, and that toxin may be a product that's produced by bacteria. So for instance, botulism, that's food poisoning, because what's happening here is you're consuming that botulinum toxin, and that is what's causing the problems. The actual bacteria that made it is not going to pass through your stomach. It's not going to survive that trip. It's not going to be living bacteria that's causing you issues. It's the metabolic waste products that they contaminated your food with. Staphylococcus aureus, if it's found in food, it can produce some waste products as well that are toxic. So a salmonella infection is an actual infection. It's active bacteria that are damaging cells within your system. Salmonella will attack the epithelial cells that line the intestine, and they cause diarrhea and abdominal cramps and nausea and fever and vomiting. It tends to be fairly short-lived, but it can be quite intense. So they manage to get through the mucus and find their way to the epithelial cells, and that's what you're seeing here. So those long finger-like structures are microvilli on the cells that line the small intestine. And these bacteria have attached to the epithelial cells and then entered the epithelial cells. And a lot like cholera, they interfere with the movement of water and ions across the membrane. And water moves out of the cells and moves into the lumen of the small intestine, and that causes the diarrhea. Traveler's diarrhea. there are lots of organisms that can potentially cause diarrhea, and traveler's diarrhea or backpacker's diarrhea is kind of a catch-all term for some sort of diarrhea that you picked up, usually in a foreign country and usually as a result of contaminated water. So typically it's someone that goes to a country where there is a microbe living in the water that they are not used to, don't have any immunity to, and it causes quite often quite nasty diarrhea, but again, it's something that you recover from fairly quickly in most cases. This can be caused by a lot of things, like I said. Usually it's caused by bacteria, so in about 50% or more of cases it's bacterial. Salmonella, strains of that can cause it, strains of E. coli can cause it, but there are other causes as well. So there are protozoan or protist causes. Giardia is one of them, Cryptosporidium. What else do we have? There are some amoeba, some ontamoeba that can cause these conditions, and there is one species of ciliate. Ciliates include paramecia. So there's one species that's very similar to paramecia that can cause these sorts of symptoms as well. And yeah, this is countries where traveler's diarrhea, again caused by a multitude of different things, are commonly reported in travelers. The liver is an accessory organ of the digestive system. So it produces bile, which contains bile salts that are emulsifiers. So bile is produced in the liver, stored in the gallbladder, and then it's dumped into the duodenum, where it's used to emulsify fats so that they can be broken down. The liver also receives blood flow from the digestive system. So any nourishment, any nutrients that are collected from the digestive system pass into the hepatic portal system and then go to the liver first, where they're processed before that material is released into general circulation. Hepatitis is an inflammation of the liver. So whenever you see HEPA, that has to do with the liver. If you have damage to the liver, a number of different things can happen. One of the most obvious is jaundice. Jaundice is where your skin and the whites of your eyes become yellow. We talked about this before when we talked about yellow fever, for instance. But what's happening there is your liver plays a number of important roles. One of them is to break down hemoglobin and recycle it. And if it's not doing that job because it's being damaged, one of the intermediate products of that breakdown, which is yellow in color, will start to build up in your tissues. Hepatitis can be caused by a number of different things. It can be caused by bacteria, viruses, multicellular parasites like liver flukes, things like that. But when most people think of hepatitis, they think of viral hepatitis. And there are several different forms of viral hepatitis. Type A is pretty common, and it's caused by unsanitary water. Type B and most of the other types are caused by blood transfer. So it could be from needle use, so you're using unclean needles. It can be spread sexually in some cases as well. Some of these viruses are rather interesting. D is quite fascinating in that it's a much reduced virus. It's a very small virus, and it actually can't do everything it needs to do on its own. In fact, D will piggyback on B. So B has to be actively reproducing in a cell in order for D to be able to reproduce. D on its own actually doesn't have enough genetic information to code for everything that it needs to have to trick the cells into replicating it. So if you have hepatitis B, you will sometimes have hepatitis D piggybacking along with that virus. Hepatitis can lead to liver cancer. So viral hepatitis, again, we've got viruses here that are inserting genetic information. That genetic information can be incorporated into your genetic information. That might interrupt a gene that codes for something that regulates the cell cycle. This is hepatitis B, and I chose this one as an example because it is quite prevalent and it can be quite damaging. It's an enveloped double-stranded DNA virus with a rather long incubation period, so it may be quite a while before you realize that you have this. And it's transmitted in a fashion that's very similar to how HIV is transmitted. So it can be transmitted sexually, through needle use, through contaminated blood. And what it does is it infects the liver, and it will, of course, destroy liver cells as it reproduces, and that can cause liver failure. And as I mentioned, the liver does so many things. It's involved in storing a lot of nutrients and vitamins. It's involved in the recycling of hemoglobin in red blood cells. It's involved in screening all of the nutrients that you absorb through your digestive system. It does a lot of stuff. So if you have a problem with your liver, that of course is pretty serious. There is a vaccine that's available, but if you have hepatitis B, at this point there really isn't an effective treatment. And finally, for the digestive system, I won't go over these in detail again because we did go over them in quite a bit of detail previously, but we have quite a few multicellular parasites, so animals that can live in the digestive system as well. So remember, we have tapeworms that will latch on to the lining of the intestine. They're very, very thin and flat, and they'll soak up the pre-digested food from your intestine and live off of that. We have hookworms that can latch on to the lining of the intestine, and they are actually going to feed on blood. So they're going to feed on blood from the tissue that they're attached to. We have Ascaris, which is a roundworm that lives in the intestine, and it will be soaking up pre-digested food, a lot like tapeworms. We have pinworms that live in the rectum and come out into the anus to lay their eggs. Yeah, just make sure you know all of the life cycles for these different forms that we've talked about. Infections of the nervous system. So we have two broad parts to the nervous system. We have the central nervous system, which is the brain and spinal cord, and then the finer nerves that radiate off of that are the peripheral nerves of the peripheral nervous system. The central nervous system is protected by three tissue layers known as meninges. So we have the dura mater, the pia mater, and in between that we have the arachnoid mater. And these tissues cover the brain, and they're continuous with the same coverings that cover the spinal cord. Meningitis is an infection of the meninges, usually by bacteria, but it can be by fungi or virus as well. So remember, inflammation means an increased amount of blood flow to an area. And there are fine blood vessels within these tissue layers. And you can see them on that photograph here. So that's a photograph of the brain. Now the kind of chunky stuff you can see growing on top, that cream colored stuff, that's bacteria. That shouldn't be there, of course. Increased blood flow to the meninges has resulted in this swelling that has put pressure on the brain and is damaging the tissues of the brain. Pathogens that cause meningitis, they can enter into the cerebrospinal fluid. That's the fluid that surrounds the brain and spinal cord. They can get there due to injury, like a skull fracture, or it can be due to a wound that introduces pathogens to the peripheral nerves, and then they can travel along the peripheral nerves and through the lymphatic system and eventually get to the cerebrospinal fluid that way. Symptoms from meningitis: vomiting, fever, pain, headache, disorientation, rashes, coma. Quite a few different things can occur as a result of meningitis, and it can of course be very, very serious. Leprosy is an ancient disease that has plagued humans for quite a long time. This is caused by a bacterium, and it's a bacterium that is very slow growing, and it can take 5 or more years before symptoms appear. In fact, it can take 20 years before symptoms appear in some cases. So it infects peripheral nerves and also mucosa. In fact, one of the first symptoms is that you have a runny nose. But it does get into the peripheral nerves. It damages them, so you can lose sensation in the skin. So you can lose sensation for pain and temperature and so on. And then if it's left untreated for long enough, it can result in deformities of the skin and even the loss of fingers and toes and other appendages like your ears and your nose. And that's due to damage within the skin that allows other opportunistic infectious bacteria to gain a foothold and damage the tissues further. So in the past, people were shunned that had leprosy. And there were leper colonies in many parts of the world where they isolated these people from each other. It's something that can be treated. So it can be treated with a cocktail of drugs. So it's a multi-drug approach to treating leprosy. And in fact, it's not terribly contagious. Some other bacteria that can impact the nervous system. We've got tetanus, which is caused by Clostridium tetani. And this is a bacterium that does actually grow in a wound. So if you get a deep wound, this bacterium can grow at the bottom of that wound because that is an anaerobic environment. It grows very slowly, but it releases a toxin, and as we talked about, that will inhibit the release of inhibitory neurotransmitters, and that causes your muscles to go into sustained contraction or tetanus. Now, kind of the myth is that you get this from rusty nails. It actually has nothing to do with rust. It's not necessarily associated with rust. It is associated with anaerobic environments, and if you've got a nail in soil, it's a perfect delivery system because the nail punctures your skin, causes a deep wound, and that will introduce the bacterium. But it's not associated with rust specifically. If you have a deep wound, it's important to clear it fully, clean it out fully, and also using peroxide is quite a good way to fight tetanus because peroxide, when it reacts with tissues, it will release oxygen, and even if the peroxide itself doesn't kill the tetanus bacteria, the oxygen released by the peroxide might. Now we also have botulism, and remember, botulism is not the result of an active infection. You don't have that bacteria living in your body if you have botulism. Instead, you have consumed a food item that contains the waste, the very, very toxic waste of Clostridium botulinum. Polio we talked about before as well. So polio in most people is not life-threatening. In fact, in most people it's asymptomatic, but it can cause paralysis in some individuals. Rabies, if you recall, was first studied by Louis Pasteur. He showed that he could take saliva from an affected animal, take that saliva, filter it, take that filtered saliva, inject it into another animal, and that animal got rabies. So he showed that there was something smaller than bacteria that could cause infection. And this is before we had electron microscopes or any of the tools we use today to analyze viruses. So the rabies virus is single-stranded RNA, non-enveloped. It has this rather complex protein coat to it. It actually looks like a bullet when you see it under the electron microscope. It's got zoonotic transmission. That means it's typically transmitted to humans from other animals, other mammals. We've got West Nile virus. So this is a single-stranded RNA enveloped virus that affects birds, but can be spread to humans, and it affects our nervous system. It can cause swelling of nervous tissues. In most people, it's not harmful, but it can occasionally be quite harmful. About 1% of people who contract it can have rather serious symptoms. A number of nervous system infections can cause what's called encephalitis. Encephala refers to the brain, itis again, and inflammation of. So we're talking about swelling of the brain or pressure being put on the brain by cerebrospinal fluid. And as you can imagine, that's not a good thing. So if this is going to affect the functioning of the brainstem, for instance, that could shut down your breathing and your heart rate. So there's several things that can affect the brain and cause encephalitis. One is trypanosoma, which, if you remember, is a single-celled eukaryote. It's a protist, it's a protozoan. And African sleeping sickness and Chagas disease, those are caused by members of this genus. We have the rabies virus that we just talked about. It causes encephalitis. It causes the brain to expand. West Nile virus does this as well. Prions cause infections of the brain. They can actually punch holes in the brain as well. Now, what's kind of interesting about rabies is it's one of the few diseases that you can fight with a vaccine after you've acquired it. So we've got a picture here, someone's being bit by a rabid animal, and what's going to happen is the virus, which is in the saliva, is going to penetrate deep into the skin and make its way towards a peripheral nerve. So the peripheral nerves come up into the dermis of the skin. And then the virus will infect a peripheral nerve cell, replicate inside the nerve cell, and move on to the next nerve cell. And it will move along the peripheral nerves, eventually getting to the central nervous system and eventually getting to the brain. That takes a long time. So what you can do is, if you suspect someone has been bitten by a rabid animal, you can give them a series of injections with antigens from the rabies virus. The immune system will now have a lot of these antigens floating around. It will launch an immune response. It'll make lots of antibodies. And because this virus grows and replicates and moves towards the brain very slowly, your immune system will have that big head start that it needs to start fighting this infection. We also have some multicellular parasite infections of the nervous system. Remember, we talked about neurocystocercosis, which is where you have larval tapeworm living in little cysts. Normally they live in muscle, but they can live just about anywhere, including nervous tissue. So here we've got the cysts of the pork tapeworm. We've got a little larval pork tapeworm living in brain tissue. And as it grows inside that cyst, it's going to destroy a pocket of brain tissue. And prions. Remember, prions are infectious proteins. They're not alive, of course. They're even simpler than viruses. They're proteins that will grab on to similar proteins and change their shape so that they match them. So we have Creutzfeldt-Jakob disease, and its analog in cows, which is mad cow disease. They may or may not be the exact same thing. There's still some debate about that. But it's quite interesting in that these prions can be transmitted as infectious agents if you consume contaminated tissue that contains the prions. You can pick them up that way. Remember that prions are very, very difficult to denature and destroy. So they can pass through the stomach, they can survive cooking, that sort of thing. And it's also possible to pick up the malformed form of the protein, the prion form of the protein, genetically. You can have genes that code for this misshapen protein. And the protein is normally found in its natural state on the surface of nerve cells. And what's happening here is the malformed version is destroying the normal version, and that is adversely affecting nervous function. So last but certainly not least, we have infections of the circulatory system. Now, once a pathogen gets into the circulatory system, it can move anywhere in the body, and it will give rise to a systemic infection. Remember, systemic is system-wide or body-wide. Some examples of bacterial infections we've talked about that can be circulatory in nature would be the plague. So remember, that's caused by a bacterium known as Yersinia pestis. Can be spread from person to person by fleas, and the reservoir is typically rodents. Remember that this comes in a few different forms. So we can have bubonic plague, where it's restricted to the lymphatic system, and we get swollen lymphatic areas that produce these bumps known as buboes. We can have pneumonic plague, where it's an infection of the lung, and in that case you can actually spread it by respiratory droplets. But we can also have septicemic plague, and that's where the bacterium has entered into the blood, and that is the most serious form of plague. Lyme disease. So Lyme disease is caused by a spirochete bacteria. The reservoir is mice and deer and other mammals, and it's spread by the deer tick. So again, plague can be an infection of the circulatory system, but it isn't always. Lyme disease is a circulatory system infection, and it is a systemic infection. If we have bacteria in the blood, this is known as bacteremia. So that's a bacterial infection of the blood. So bacteremia, again, an infection of the blood, can start off with a local bacterial infection, maybe just in a deep wound or something like that, that then spreads from that area through capillary beds and into larger blood vessels. This can bring about something known as sepsis, and this is a very vigorous, overwhelming immune response. So it's a body-wide inflammatory response that not only damages the pathogen, but may damage your own tissues as well, and can lead to organ failure in some cases. Toxemia is the name for the presence of toxins in the blood. So notice when you have emia at the end of a word, you're talking about something in blood. We can have toxins being released from gram-negative bacteria over a very short period of time, if we have lots of gram-negative bacteria dying over a short period of time. And this causes something known as septic shock. So imagine that you have a systemic infection with gram-negative bacteria. Your immune system does a really good job, or maybe you take some antibiotics that do a really good job, and all of a sudden you have a whole lot of gram-negative bacteria dying. Well, recall that they have endotoxins. Those are toxins that are found in the cell wall, generally in the outer membrane. Those are going to be released when all these bacteria die, and now you might have a huge problem on your hands, because these toxins can be very damaging to tissues, and they can cause organ failure and all sorts of nasty things. This can be something that is life-threatening. Systemic viral infections would include infectious mononucleosis, commonly known as mono, or the kissing disease, because it's spread by saliva. This is caused by the Epstein-Barr virus, which is one of the herpes group of viruses. Something that will spread throughout the body, generally not life-threatening. It can cause some long-lasting effects, although that's quite rare, but it produces fever, sore throat, swollen lymph nodes, and just general fatigue. So treating this, there is no direct treatment. It just involves treating the symptoms and keeping the person as comfortable as possible. But most people have antibodies in their body against it, so it suggests that most people have been exposed to it. Many people are probably asymptomatic. Yellow fever, we talked about that in quite a bit of detail. Remember that this is a virus that's spread by mosquitoes. Monkeys can be a reservoir for this, and we talked quite a bit about yellow fever when we talked about epidemiology. Ebola. So Ebola is an incredibly virulent virus. Again, remember that means it causes a lot of damage. Fortunately, it's not that contagious. So it's not that pathogenic, but it is very virulent. So you can imagine this is a virus that people want to keep an eye on, because if it did suddenly become contagious, if it mutated in such a way that it became more contagious, it would be very, very worrisome. It causes hemorrhaging, which is bleeding. So it causes internal hemorrhaging, so blood pooling inside your body cavities. Not pleasant. Malaria is caused by a protist. So this is a protozoan pathogen. Plasmodium is the name of the pathogen. Remember that it is a member of the apicomplexa. It has an apical complex that it can use to punch holes in cells, and then it reproduces inside those cells. So it can punch holes in liver cells, and it can punch holes in red blood cells as well. Chagas disease, caused by trypanosoma, which is a blood parasite. And this parasite is spread typically by something called a kissing bug. And you're seeing a picture of it there. So this is an insect that will use this little needle-like device, proboscis, that it uses to drink fluids, to drink a bit of your blood, usually from around your lips as you're sleeping. That's how it gets its name. And I should point out that we have very similar looking insects in Canada, but they're not these kissing bugs. The ones that we have around here feed on the fluids of insects. They actually hunt other insects, or they feed on plant juices. So don't be too freaked out if you see one of these in your garden or in your house. Malaria, we talked a little bit about this when we talked about epidemiology. Again, as I said, it's caused by Plasmodium, which is a protist, has several stages. It has to pass through a mosquito to complete its life cycle. So part of the life cycle is spent in mosquito. The other part is spent in human or another primate, typically. To control this, there's a few things we could do. There are drugs that can be used to treat it if you already have it, but limiting your exposure to mosquitoes in tropical and subtropical areas is the easiest way to limit the spread of the disease. And as we talked about when we talked about epidemiology, there's been a big push to make sure that we have netting for beds supplied to the countries that need it, so that they don't have to sleep being exposed to mosquitoes. So if we can cut down the transmission of malaria between hosts, then we can cut down the reservoir of Plasmodium. And this brings us to the end of a rather long topic. Just a few quick concepts here, kind of the really broad take-home messages. It's not a bad thing to have microbes living on your body and in some parts of your body, the colon for instance. Microbiota, the common resident bacteria and other microbes, serve a useful function. Some of them will produce vitamins for you, etc., but others, even if they're not helping you directly, they compete with microbes that might cause infections. They also encourage activity of your immune system. They prime your immune system. You'll make antibodies against common proteins on these microbes to limit their growth. And those antibodies may prove to be useful against potential pathogens. So you have an ecosystem on and in your body, and these microbes are fighting it out. They're competing with each other, and that's a good thing. If you disrupt this ecosystem, this ecology, that might create problems. If you get rid of bacteria that are producing acidic conditions in certain parts of your body, then microbes that are limited by acidity may become problematic. The human body is a perfect incubator for lots of microbes. As I've mentioned before, you have more bacteria living in your intestine than you have human cells. That's pretty remarkable. But you also have many, many barriers in place to keep them where they should be for your health. Pathogens that get into our bodies and cause us problems have to enter through a portal of entry. And there's several different portals of entry. It could be something that is injected beneath the skin, maybe something that's found on a splinter, for instance. It could be something that is breathed into the respiratory system or something that's ingested. Once this pathogen is inside the body, if it's going to produce a troublesome infection, it has to evade the host defenses, the immune system, etc. Many pathogens, so some protists, some bacteria, and all viruses, will enter into the cell. And once they're in the cell, it's more difficult for your body to attack. If there's symptoms, that's generally the result of damage to cells, or perhaps an overactive response of the immune system. Now, the pathogen, if it's going to spread, has to leave the host and find another host, and it's going to leave through a portal of exit. And quite often the portal of exit is the same as the portal of entry. Pathogens can damage cells and tissues by secreting exotoxins, especially if we're talking about bacteria. Some bacteria contain endotoxins. These are integral parts of the cell wall typically, and when the cell dies, those toxins are released and it causes further damage, and that's something to be very aware of. If you kill the pathogen, you may actually be causing other side effects. And here's our terminology list. It's surprising it's not longer than this, considering the length of this topic. Here's a few good study questions to consider. And here's a few more. Might these form the basis of a question on your exam? Maybe. And finally, I thought I would end off with a few useful endings. See what I did there? So here's some Greek and Latin endings that might be quite helpful. So emia, this is something in the blood. Toxemia, we talked about that, for instance. Itis is an inflammation of something, whatever comes before it. Osis is an infection caused by something. Mycosis would be an infection caused by a fungus. Cyte means a cell. We've seen that a lot now. Phago means to eat, so phagocyte is a cell that eats. Cidal means to kill. If you're suicidal, hopefully you're not, that means to kill the self, the sui. Homicidal, that means to kill a man. It's a bit sexist, but to kill a human. Static means no change or growth. So bacteriostatic means that it's going to limit the growth of bacteria but not kill them outright. Always pay attention as you go on in your schooling and career to endings like this in Latin and Greek words. They will really help you out. Once you see them and identify them, even if you've never seen a word before, you'll hopefully be able to figure out what it means. So hopefully you found this course interesting. It is unusual the last year and a half with COVID and not having face-to-face classes. So you've had a different experience than most students, but hopefully it wasn't a bad experience. Good luck. And as I've mentioned, I am going to have a review session, so stay tuned for that. And I will have a very short little PowerPoint that kind of goes over COVID in a bit more detail, just to kind of have it all in one place. It's all stuff we've talked about before for the most part, but I just thought it would be nice, given that this is an unusual year, to address what's made it so unusual and summarize all that stuff.